Okay, so welcome to this next video in the playlist on the heart. In this video, what we're going to do is continue our discussion of pathological cardiac hypertrophy. And this video is a follow-up to my video on the sarcoplasmic reticulum and pathological cardiac hypertrophy. So I would highly recommend that you watch that prior to watching this. In this video, what we're going to talk about is how SERCA2 function is reduced in cardiac hypertrophy. Okay, so SERCA2 function is what we're going to discuss. Right. So the way I'm going to structure this video is I'm going to start off with a summary of everything that we saw in the video on the sarcoplasmic reticulum and pathological cardiac hypertrophy. So basically what we saw was that if the heart for some reason is struggling to pump the blood out of the left ventricle, let's say, into the aorta, then what starts to happen is the cardiomyocytes of the wall of the heart start to undergo pathological cardiac hypertrophy, also known as pathological remodeling. And what happens in pathological cardiac hypertrophy is that if this is a cardiomyocyte, what happens is they grow, basically. They become much thicker and they become a little bit longer, but basically the change in thickness dwarfs the change in length. So you see the cardiomyocytes becoming much fatter, basically. Okay, and this form of hypertrophy is known as concentric hypertrophy. And initially, the cardiomyocytes hypertrophying like this actually helps with cardiac function, because initially they become stronger. The capability of this concentrically hypertrophied cardiomyocyte is actually initially greater, basically, than the initial cardiomyocyte. So its ability to generate force is greater. Okay. However, the problem is that it's short-lived. Initially, it will be able to generate a greater amount of force. However, later on, the amount of force that this concentric hypertrophied cardiomyocyte is capable of generating will actually decrease to lower than the level of force that this pre-hypertrophy cardiomyocyte was capable of generating. So it's sort of like a short-lived solution, works for a while, but then things go wrong. And we wanted to study why, what actually has happened, what's changed within these concentric hypertrophied cardiomyocytes that means that their ability to generate force is reduced. So basically what we found was that if you look at how much calcium is released on each beat of the heart, so if you look at how much calcium is actually released into the cytoplasm of the cell when the cardiomyocyte undergoes an action potential, that is actually reduced compared to the previous unhypertrophied cardiomyocyte. Okay, and we dug a little deeper to actually explore the reason for this, and we found that it was because the sarcoplasmic reticulum had less calcium stored within it. And then what we saw is that the reason for this was some sort of dysfunction in SERCA2. So SERCA2's function was reduced. In this video, what we want to look at is why is SERCA2's function reduced. Okay, so the first thing to say is that there's a very simple explanation initially. One of the explanations for why SERCA2's function is reduced is that you just don't have enough of it. So, let me remind you of what SERCA2 is. So, within the cardiomyocyte, you have the sarcoplasmic reticulum. So, let's draw the sarcoplasmic reticulum within this cardiomyocyte here. So, here's our sarcoplasmic reticulum, and it will have a huge number of these processes coming off it, which will be forming synapses with the T-tubules that may well come in. So let's draw a T-tubule here. Let's draw maybe another T-tubule here. Okay, and it will continue to go off and form many different calcium synapses. Right, so the sarcoplasmic reticulum shown here is this intracellular organelle, which is a store of calcium. So there's a lot of calcium stored within this sarcoplasmic reticulum. Now, when the when this cardiomyocyte undergoes an action potential, what will happen is at all of these calcium synapses, as they're called, these interfaces between the plasma membrane and the membrane of the sarcoplasmic reticulum, these are called calcium synapses. When the plasma membrane undergoes an action potential, the plasma membrane will allow calcium to come into the cardiomyocyte, into the cytoplasm from the extracellular space. And this calcium that comes in from the extracellular fluid will then trigger the calcium release from the sarcoplasmic reticulum, so-called calcium-induced calcium release. All of the calcium that is released from all of these calcium synapses is going to sum together to create a signal that's known as a calcium tide. Okay? So when calcium goes up globally in the cell cytoplasm, because all of these little calcium sparks, as they're called, are summing together. So remember, when you release calcium from the SR, from one of these calcium synapses, that release of calcium, the rise in calcium nearby the calcium synapse, is known as a calcium spark. So you're getting calcium sparks occurring at every single one of these calcium synapses, and that, when they all sum together, is what's known as a calcium tide, when the calcium level within the cytoplasm of the whole cell is going up. Okay, so you get this calcium tide, and that causes the cell to contract. Okay, but you have to return the calcium into the SR so that on the next beat of the heart, when this cardiomyocyte next undergoes an action potential, you can do the whole thing again. Okay, so you have pumps on the membrane of the SR, okay, known as SERCA pumps. Okay, and I've drawn one here, but in fact you'll have many of these, so lots of these SERCA pumps. And SERCA stands for Sarco/Endoplasmic Reticulum Calcium ATPase. So let me write this down. Sarco/Endoplasmic Reticulum Calcium ATPase. Okay? And you take the initials of that to get the SERCA name. Whoops, what am I doing? I'll have to. There's no reason I've put that A as a capital. It's just because for some reason my brain isn't working properly. It decided to put a capital A there. It was because there should be a capital A on ATPase like this. So I was just getting a little ahead of myself. Right. So Sarco/Endoplasmic Calcium ATPase. Okay. And these pumps, basically, if I draw one of them out bigger down here, they pump calcium back into the lumen of the SR. So they pump 2 calcium ions into the lumen of the SR in exchange for 3 protons leaving the lumen of the SR. So every time the SERCA undertakes its activity, what it will do is it will pump 2 calcium ions into the lumen of the SR. So this is the SR lumen side, and it will pump 3 protons out of the lumen of the SR into the cytoplasm. Okay, and when it does that, it requires energy to do that. So the energy comes from the hydrolysis of ATP, hence the name Sarco/Endoplasmic Reticulum Calcium ATPase. So it's a enzyme which breaks down or hydrolyzes ATP to adenosine diphosphate ADP and an inorganic phosphate molecule PI. Okay, so these are pumps which actively transport calcium from the cytoplasm back into the SR lumen. Now, the function of these pumps, and specifically, let me just add one more little bit of information. There are different forms of the proteins that make up this pump, and if you therefore have differing isoforms of SERCA, and the SERCA which is found in cardiac muscle cells is the SERCA2 pump. So specifically it's SERCA2 that you find in cardiomyocytes. Okay, so basically in these concentrically hypertrophied cells, you have lower SERCA function. Okay, lower function of SERCA2. So the ability to pump calcium from the cytoplasm into the lumen of the SR is reduced. Now, what could cause this reduced function in SERCA2? Well, if you simply had fewer pumps in the membrane of the sarcoplasmic reticulum, that would cause reduced function. So in fact, you find that you have underexpression of SERCA2. So simply you don't have enough of them in the membrane of the SR in cells which have undergone concentric hypertrophy. Okay, so that's one of the first causes of this SERCA2 underfunction, that you simply have too few of them. Now, a nice correlate of this is we've made this prediction, so we've observed maybe that the number of SERCA pumps is too little in the membrane of the SR. So now what we would predict is that if you raise the level of SERCA pumps in the membrane of the SR, that the calcium spikes will be returned to normal, because remember, the problem in this concentric hypertrophied cardiomyocyte is that because the SERCA pump is underfunctioning, the amount of calcium in the SR lumen is too small. So when this cardiomyocyte undergoes an action potential, the amount of calcium that's released is too small, and therefore you don't recruit as many sarcomeres, you don't generate as much force. Okay, so that's why we think this concentric hypertrophied cardiomyocyte is less capable of producing force. So if we replace the SERCA2 pumps, that should return the size of the calcium spikes, and therefore the amount of force that the cardiomyocyte is capable of generating. So we can do this basically by adenoviral gene therapy, which I'll describe to you in the next video.